Hello, everybody. Hope you're all having a good day. We're going to start into the cells unit now. So, just so we did biochemistry and we did evolution for the first quarter. We're going to do cells and metabolism the second quarter. And so, cells probably pretty self-explanatory. A lot of stuff you guys have gone through in regular pre-AP bio before, with of course a few more layers of details and information added in, a few new sections probably. And then we're going to go into metabolism, which is cellular respiration and photosynthesis and some enzyme stuff. So actually, hang on one second. I forgot I wanted to go over some of those cell answers with you on that discussion that will give you an idea of how many cells there are and all sorts of things like that. You can probably find them in different sources and find different estimates and stuff like that. Many of mine are just ranges. But for the first one, how many cells are in the human body? If you said million or billion, you underbid. Okay, it's definitely in the trillions, and the range I have is 10 to 100 trillion, with there being about 4 trillion of solid mass. Okay. On number two, how many types of cells are there in the human body? Last I checked, there were 210 types of cells. Okay, so tons of cell types as well. How many neurons are in the adult human brain? Quite a few. You need to be in the billions with your answer. It's about 100 billion neurons, and within each around each neuron, there are between 10 and 50 glial cells, Glial, glial cells, and those are there to support and nourish the neuron because they're so busy and active. Okay. How many bacterial cells are in your body? You might be shocked to know that it is between 40 and 50 trillion. So you are riddled with bacteria. I'm sorry if you're a germaphobe. There is no escaping the bacteria. Now, not all of that bacteria is bad. Some of that bacteria is very good for us and it helps us. So, but we do have a lot. And I don't remember, I think I asked you the DNA question as well. All of your cells have the same DNA. You would think that would be true, and it should be true, but it's really not. We do have mutations. We do have these things called SNPs, which are single nucleotide polymorphisms. So we do have, some slight differences here and there. And what types of things do scientists do to learn about cells? I think several of you mentioned microscopes. And there are several different types, which I'll talk about today during the notes about the scopes. And then experiments. There's tons of different experiments people do, whether it's tissue culture stuff or genetic stuff. Lots of stuff to do with cells if you're getting at the DNA, or you can do different experiments with the proteins in the cells, track them, and all of that. An easy thing to do with cells is count them. A lot of times scientists learn a lot by counting them, right? Like you go to the hospital and they take your blood and they do a cell count and they see, are your white blood cells elevated or your white blood cells dropping, that sort of thing. So a lot of times just counting certain types of cells give us clues as to what's going on. Okay, so I think that's about all I asked you on the discussion. I thought I'd share those here. So if we get into our notes here, here are the big questions. Why does life require cells? How are they organized? What's the advantage of the organelles? And how does the interaction of all these components allow for life processes? Okay, so lots of pictures, no matter what it is, right? No matter what living thing it is. If it's living, it's made of cells. Okay, so the DNA is in our nucleus, and it's going to contain the instructions for telling the cells to differentiate into certain things and how to organize and what the proteins to do, and that's why all these living things look differently, is because the instructions vary just a little bit. So quick info on slide history. Cells were discovered in the 1600s, and we call the study of cells cytology. Anton van Leeuwenhoek He's associated with the first microscope. He actually didn't build the first microscope, um, but he improved on it, and was for a long time was given credit for the first microscope. He really just improved the lenses. Um, and kind of made it better. And so, um, he is Dutch. If you're Dutch, you're from Holland or the Netherlands, so that's where he's from. The other guy over here, Robert Hooke, is English, and he was the guy that ended up giving them the name cells, because he thought when he looked through, they were looking at plant cells, and so you can see they kind of look like little bricks up there, all stacked up, and it reminded him of the rooms that the monks stay in at the monasteries, and so those were called cells. So he named these cells. They kind of like dorm rooms, little square rooms all in a line. Okay. So how do we know what's in a cell? Well, most times we use microscopes, right? Light microscopes can go up to 1,000 or 2,000 magnification. The microscopes that most of the Zumwalt schools have go up to about 400. And that's still pretty good. You can see quite a lot on high power with a good light microscope. Nothing you need to memorize on this, you know, uh, table at all, but just kind of want to show you there are a bunch of different types of microscopes here that are used for all different sorts of reasons. Some that you can stain, and some that you use fluorescence with, and some use lasers and all sorts of stuff. But these are all different types of light microscopes that can be used. The ones that we really learned from a lot more recently are the electron microscopes, and a couple of them I want you to know. The SEM stands for scanning electron microscope, and you can relate scanning to surface, the two S's. Okay, so if you look at those images up top, you can see it's looking at the surface of the either organism or cells or whatever. The transmission one at the bottom, transmission electron microscope, is the TEM, and that one allows us to look inside. We make thin little cross sections of organelles or cells or whatever, and we can then see to the inside of the cells. As you can tell with the pictures on the bottom, you can see some organelles and stuff like that going on, and that's because of a transmission electron microscope. Let me pause for 1 second here. Okay, sorry about that. Just need a sip of water. If we think about cell size, here's a good chart right here. If you look at, everybody thinks, cells are pretty small, right? But they're actually, over here, and there's lots of things smaller than a cell, right? There's all the organelles and viruses and bacteria and ribosomes and, all the organic molecules, proteins, lipids, water molecules, atoms, all those things are smaller. Cells are just a little bit smaller than what we can see with the unaided human eye. So they are small, but not quite as small as some other things. So just a couple pictures of different cells up here. We just have a little discussion about, different cell types and stuff. So you have red blood cells here in the upper left, and then on the big picture on the right, you might uh, guess that would be the gametes. So that is many sperm attaching to one egg. And you can see the size difference between the egg and the sperm. Someone in class said that was the flying spaghetti monster, which is kind of appropriate. So, and then down here, you see the big oval. You see this oval section right through here, and then you see all these guys kind of attached to this oval, and maybe you have some guesses about that, maybe not. But all those guys are there. So what's going on with that one is the oval guy, the big guy, is like a bacterial cell, and all those little structures that look like trees or whatever that are attached to it, those are viruses. And viruses are kind of attacking that cell right there. And the way viruses work is they actually inject their DNA into a cell, and that DNA then takes over, it gets incorporated and kind of takes over the machinery of the cell. It kind of brainwashes the cell. And many of them then turn the cells into virus making machines. So then it makes more virus. And that's how viruses kind of spread throughout the body and stuff like that. So just a little bit of virus info, kind of pertinent with everything that's going on. So what units do scientists use to measure cells? Centimeters would be too big, right? Millimeters, think about the really tiny lines on a ruler, still too big. We have microns or micrometers. We just abbreviate it or call it microns a lot of times. A micron is if you took a millimeter, remember the skinniest part, the skinniest lines on the ruler, the space in between them, and if you divided that by 1,000, you would get a 1 micrometer. Okay, so it's 1,000th of a millimeter. And then if you go to nanometer, you take that micron and you divide that by 1,000, and you will get a nanometer. Okay, so super small. So which one do you think scientists use to measure cells? You think it's microns or you think it's nanometers? Well, based on the previous slide where I showed you some of the sizes of other things, you might have a guess. Hopefully you said microns. Okay, so cells are usually between 50 to 200 microns. Here's an example. A lot of times they give you the little legend down there in the corner that shows you like how much distance is 50 microns, and you can kind of estimate a cell size in a picture from a book or on a PowerPoint or whatever. And then if you look, actually let me back up. Notice the funky little symbol here. Just kind of wanted to make note of this. This guy right here. Okay, so that when you see that, which kind of looks like a U with a really long tail on the front side, next to the M, that's just the symbol for micrometer. I think it's a Greek letter. I can't remember actually which one, but that's just so you know you're looking at micrometers. Nanometers are actually used to measure the smaller things, atoms and molecules and viruses, and things like proteins, which are 5 to 10 nanometers in size. Atoms are super small, obviously. Okay, so all those types of things we measured in nanometers. Cells is typically microns. Okay, so question for you: why are cells so small? Okay, why are cells so small? Why do I look so young? Because that picture was taken several years ago. Why don't cells get this big? Okay, why don't cells get really huge? Why aren't we made of, maybe 10 cells the size of basketballs? Okay, or why aren't we made of 300 ping pong ball size cells? Okay, so why is that? Why do cells have to stay so small? Why don't they grow big? There's a few answers for that, and obviously if this was live, we would have a big discussion about this, and I would ask you for your feedback. But in this format, I'll just keep trucking along here. So a couple things to keep in mind. If you look at a cell as a cube, you look at what the surface area of the cube is, right? The outside surface of the cube. And then you look at the volume, right? Those are the two things we can really look at as far as the space and the surface of the cube. And so if you look at 3 different sizes, a 1 centimeter, a 2 centimeter, and a 3 centimeter cube, and then do a little simple math, you can learn some things. So the surface area, if I grab my pen here. So the surface area of the 1 centimeter cube down here says 6 centimeters squared. So for you math people, how do you get the surface area of a cube? Well, you take the area of one side. Okay, you take the area of one side and then you multiply it by 6 because there's 6 sides, right? So length times width. So 1 times 1, okay, gives us the area, and then we have 6 sides, so we get the surface area being 6 right here. And over here, we take 2 times 2, which is 4, times 6 is 24. And then over here, we have 3 times 3, which is 9, times 6, which is 54, right? And so that's how we get those surface area numbers. And then volume, length times width times height, right? So you get a 1, and you get an 8, and you get a 27. And then if we do a little reducing and we make our ratios, we get 6 to 1, and we get 3 to 1, and we get 2 to 1, okay? So if you look, what's the relationship between the size of the cube and the ratio of surface area to volume? Okay, this is the important ratio, the surface area to volume ratio. And so if you look at them, as the cube gets bigger, right? As the cube gets bigger going this way, then the ratios tend to shrink going this way, right? So let go. So my question now is, does the surface area or the volume increase faster? Okay, does the surface area or the volume? Look at the numbers there from the small cube to the big cube. and they're not going up at the same rate. The surface area and the volume are not increasing at the same rate. Which one is increasing faster? So pause the video if you need to, look at those numbers, and try to figure out, is it the surface area or the volume? So hopefully you took a look at that, and you should notice that on the surface area, it starts at 6, right? 6 centimeters cubed, and if you go way down here, it's at 54. Well, how many times did that increase by? Well, 6 to 54, right? That is a 9x increase, right? A 9 times increase. If we look at volume, it goes from 1 to 27, right? Well, that's pretty easy math to do. That's a 27x increase. So the volume is the one that's increasing faster, right? And when you think about that, if the volume's increasing faster than the outside, which is the surface, that's going to create a problem, right? If you get bigger cells, that's going to create a problem. Think about a balloon. What happens if you put too much volume in a balloon? What happens to your surface area? It gets stretched really, really thin and then ultimately pops, right? So while cells may not work quite the same, the membrane, the surface area is the cell membrane. If it stretches like that, it's going to become compromised, right? And while some cells do burst under some conditions, it would certainly become leaky. It would certainly, not be able to manage what's coming in and what's coming out. And the cell membrane is there to maintain homeostasis. So that would be a huge problem. Okay. And there's some other problems that we'll talk about as well. So, whether you're a person or whether you're a whale, it makes sense to have a lot of really small cells make us up so that our surface area to volume ratio stays good. We can get more nutrients in that way and more waste out, and they're just much more efficient. They operate much more efficiently at a smaller size than they do at a bigger size. So we get to the cell theory. So you guys have probably seen this before, some basic tenets about cells. All things are made of cells. All living things are made of cells. Cells are the basic unit of life, and cells come only from other cells, right? And that one should have a question going off in your head, right? Especially given the unit we just came out of. Cells come only from other cells, so the question you should, maybe pop into your head there is, well, then where'd the first one come from? But you guys know how to deal with that question now. You guys know about protobionts, and you know about how the organic molecules might have accumulated and turned into protobionts and led to cells and so on and so forth. So now you have a better answer, a better reason to give someone on that question. So here's Ron Burgundy, right? Anchorman. So, and he says that structure is related to function always, right? Structure is always related to function, and that's been one of our main themes since the beginning of the year. So just pointing out a couple of cell types here. We have neurons in the upper right, and then right below that, the yellowish ones are adipose cells, which are better known as fat. You have the one on the bottom right, the pink with all the lines going through there is striated muscle. And then you have the bottom left one there being some epithelial tissue. And those of you in anatomy are doing tissues right now, you should hopefully know that that's columnar. Okay, so hopefully you've started in on your anatomy tissues and you know a couple of these. All right, moving on to some cell types here. So prokaryotic cell structure, okay? Prokaryotes. Prokaryotes do not have a nucleus, okay? And they do not have membrane-bound organelles, okay? These are your basic type cells, mainly bacteria, right? Prokaryotic cells are all of your bacteria types, and there is some blue-green algae that's also prokaryotic. And then if we look at some structures that are that go into a prokaryotic cell, they still do have some structures in there. While they don't really have membrane-bound organelles, they do have some structures. So they have cytoplasm, right, which is like the gel inside of the cells. They do not have a nucleus. We tend to refer to this as a nucleoid. It's a area without a membrane that is where the DNA is located. Okay, so they do have DNA. They do have ribosomes. And if they have ribosomes, what can they do? Ribosomes make proteins. So bacteria make proteins in order to function. Okay, so you see that they have a cell membrane, um, the inner layer there, cell membrane, plasma membrane, same thing. Okay, those two are used interchangeably. You might also hear fluid mosaic model or phospholipid bilayer, all the same stuff, all the membrane. Okay. Remember, bacteria have cell walls, right? You should be able to tell me what molecule makes up the cell wall in a bacteria because we did that in the biochem unit. It's a carbohydrate. It's a polysaccharide. Do you remember which one? If not, you may want to go back and check because you do have a final exam coming up in a couple of months here. Sometimes there's even something called a capsule, which could even be a thicker barrier around that, and that's actually why you have to cook your food to certain temperatures, right? You have to cook your beef and your chicken and certain foods to those kind of temperatures because they might have had bacteria in them, and so you have to cook it to a temperature in order to crack that capsule open and then let the heat get in there and kill the bacteria, so we don't get sick. You don't get like salmonella from chicken or something like that. So this is just another diagram, virtually the same thing, but a micrograph, a picture from a microscope. There's just some lines there for you to have some fun with and try to name the structures. A couple of them, given the picture, you could probably interchange. It looks like they're pointing at the same thing, but as long as you name the two different things, you'd be fine. Couple things about the DNA in there. The ends of the prokaryotic DNA are connected, which means they form a loop, they form a circle. Prokaryotes, bacteria, they only have one circular chromosome. All that DNA you see in there is all linked together into one big loop. It's just folded back on each other, but it's all connected into one large circular DNA. Okay, and sometimes there are several smaller circular loops of DNA in there, and those are called plasmids. Okay, and we'll talk about plasmids more when we get to genetics, but they hold little specific pieces of code that are done for little specific jobs. Eukaryotes, moving on from prokaryotes to eukaryotes. Now we're getting into cells that are animals and plants and fungi and protists and probably more things you're even more familiar with. So eukaryotic cells are usually a little bit larger than the prokaryotic ones. They have a nucleus. They have many organelles, okay, that are usually surrounded by a membrane. Each organelle gives us a specialized function, right? Kind of like your organs. You have lungs, you have a heart, stomach, liver, spleen, all of that, and they do specific things, right? Your organs do specific things, as do the organelles for your cell. Very similar type duties there. Two major types of eukaryotic cells. You have photoautotrophic. Okay, so autotrophic, make their own food. Photo from light, so that would be plants like photosynthesis. And chemoheterotrophic, or animal-like, right? Heterotrophs have to go out and get their own food. Okay. Pro no you do. This is just a phrase I use in regular bio, and I bring it back for AP bio. If you've had me before, I'm sure you've seen it. Those of you that haven't probably are very confused. Pro no you do. Prokaryotes, no nucleus, no membrane-bound organelles. Eukaryotes do have a nucleus and membrane-bound organelles. So there you go. Pro no you do. Little simple phrase to help you remember. So why organelles? What's the big deal about organelles? Why do we need them? So organelles are really there. They give us specialization. They give us little microenvironments in order to operate differently, right? Like our stomach has a different environment than our lungs, right? Very different environment, so they can get their job done. Same kind of thing in organelles. They might have different pH or molecule concentration. I mean, all sorts of different things that they might have to have these little microenvironments for to get their jobs done. Another one is the membranes of the organelles provide surface area for reactions. All the chemical reactions that take place in our cells, I shouldn't say all, a lot of them, okay, a majority of them take place on membranes. And so the organelles having membranes gives us surface area for those reactions to take place. And so specialization is why the eukaryotes are more complex than prokaryotes. They don't have them, so they don't have the ability to have all these specialized functions. So many compartments, so many options. So here we have a cell to-do list, okay? So I have 5 things. I've tried to simplify the cell down into 5 things that it does. Processing matter. Okay, you build proteins, carbs, fats, nucleic acids. For our purposes, we're going to focus on proteins. Processing energy. Okay, you have to get and process nutrients. You have to do gas exchange. Process information. This would be cellular communication, internal and external. Homeostasis, maintain your internal environment, and then build more cells. We have to have growth and repair and reproduction of cells in order to carry on. So there's your cell to-do list, all the things that are trying to be accomplished. If I had to pick 3, I'd probably say these are the 3. Okay, so processing energy, you have to have energy for all these activities that are done by the cell. You have to get rid of the waste that's created by making the energy or transferring or processing the energy. Um, and so making proteins is huge. Proteins do all the work in the cell. We need lots of them, lots of specific functions. And then you have to be able to have, mitosis and meiosis. You have to be able to have cell division in order to carry things on as well. So we are going to look at, here's the protein to-do list, right? Proteins do a ton of different things. So this is just going to be a quick overview, hopefully a reminder of some things for you. So proteins can build molecules, digest molecules, carry out chemical reactions, provide structure, copy DNA and RNA, send and receive messages. Lots of things happening, right? So if you remember from your earlier bio days, um, this right here is the central dogma, right? DNA makes RNA makes protein. I think I've mentioned that once or twice this year already. Right here is the boundary. So that up here is the nucleus, right? This is the nucleus, and we know that because there's the DNA, and the DNA never, ever, ever leaves the nucleus, right? DNA is in the nucleus, and it's going to transfer the information into an RNA, a single-stranded molecule, and that RNA is going to go through some procedures, which we'll learn about in another unit, and it's going to then leave the nucleus, and it's going to go out here and find the ribosome right here, and that's going to end up building this protein, okay? So this is your polypeptide, your protein. So that's just a basic overview. I'm not going to ask you a lot of details on protein synthesis right now, but that's important and one big job that cells get done. So we're going to kind of look at that process from the inside out, from the beginning to end. So for the nucleus, which is where all this starts, right? Because the DNA is in there. So you have the nucleus, you have the nuclear membrane, you have the nucleoplasm, you have the nucleolus, you have the DNA in there, and you have nuclear pores. These are all the structures associated with the nucleus, and we're going to run through some of them here over the next few slides. So here are some nuclear pores for you. The nuclear membrane is 2 membrane layers thick, and if you look, there are some pores or some holes that things can go in and out of the nucleus. So maybe start thinking to yourself, what are some things that would be able to go in and out of the nucleus? Why would we need things to go in and out of the nucleus? But maybe start thinking about that. And you can see over here the pore spans the entire length of the membrane and creates a little tunnel there for things to get in or out. So this is going to follow this kind of the same pattern here where you'll see some pictures like cartoony things, and then you'll see the actual microscope picture so you can kind of see how things look. Okay. So again, the really dark nucleolus inside there. You have the double membrane, the nuclear membrane there. You have a nuclear pore. There's a hole. Here's the DNA. And remember, when DNA is unwound and it's all like spaghetti-like, and it's not that coiled, we call that chromatin. And then when the DNA, the same piece of DNA, the same piece of chromatin, coils up really tightly, we call those chromosomes. Okay? So DNA, chromatin, chromosome, it's all DNA. It matters what state it's in. Okay? Naked DNA is just the helix, right? Back all the way to stripped down DNA with just the double helix. Chromatin, it does have a little bit of coil to it. It's usually about 30 nanometers big. and we call that the 30 nanometer fiber. And then when that starts coiling and starts looking like an old school phone cord, then you start getting what we call chromosomes. And chromosomes only coil like that when we're going to divide the cell and we need to move the DNA around. So if we were going to be doing that, here's what something might look like. We do have 46 total chromosomes in the human, which would be 23 pairs. The first ones with all the numbers, can't find my cursor. So the first ones with all the numbers, right? Here's 1 through 22. Those are all called autosomes. And then you have the two sex chromosomes down here, where you have, in this case, you have XX, and in this case over here, you have XY. So you should be able to tell which one is female and which one is male, even though this one already tells you right here. XX, when you have two X's, that is female, and when you have XY, that would be male. Okay, notice the Y is a little bit shorter. We'll talk about all that when we get to genetics. This is just a kind of an overview of some stuff that's happening in the cell or involved in the cell here. So the nuclear membrane is going to protect the DNA, right? That's going to be its main job. A lot of membranes are there for protection. The nucleopore, it allows for two things to escape. RNA, which you might have thought of, right? Because the DNA gets transferred into RNA inside, and the RNA has to go out to the ribosome. So you might have thought about the RNA leaving, but the ribosomes actually have to escape. The actual protein factories start off in the nucleus, and that's because of the nucleolus. Its job is to build ribosomes. And so the ribosomes get built in the nucleolus, and then they leave the nucleus, and then eventually the RNA goes out and finds those ribosomes and makes some proteins. So here are some ribosomes. So ribosomes are generally just shown as little dots. They can show up a couple of places. They can be in the cytoplasm and be free, or they can be bound to the endoplasmic reticulum, and in particular, the rough endoplasmic reticulum, or the rough ER. Okay, ribosomes have two subunits, small and large, and they fit together kind of like a hand and glove. You can see the little site in there. It's going to fit right into, and it's going to go in there like that, and that creates a groove for the RNA to like slide through. Okay, that's going to create a groove right there for the RNA to slide through and then be read by the ribosome. Okay, and I had my old biology teacher who was really awesome, that worked at South High, whose name was Mr. Ulmer. He always said that the structure of a ribosome kind of looked like this, kind of like the hang loose sign for Hawaii. So he'd always run around saying ribosome, dude. And so maybe that'll help you remember a little bit. Who knows? Just wanted to pass it on. So ribosomes are where all proteins in the cell are constructed, the protein factory. So again, just a quick overview of protein synthesis, okay? So DNA in the nucleus copied into RNA, and that's called transcription. And then the RNA leaves the nucleus and it goes out to the ribosome, and it then the ribosome converts that code and uses tRNA and strings some amino acids together to make a protein. Okay, so just a quick reminder about the ribosome function. So the endomembrane system, okay, the endomembrane system is all of this, all of the organelles you need involving protein synthesis. Okay, so you have the nucleus, you have the ribosome, you have the ER, you have the Golgi apparatus or Golgi body, either way, and then you have vesicles. Okay, so really, if you look at a cell and you look from the inside right here, which is the nucleus, it all kind of just weaves its way, and it's all kind of connected all the way through all of this, kind of in this order right here. And that's kind of how things are organized and how it works. So we'll run through them here one by one. So again, protein synthesis, the order, the nucleus, then DNA to RNA, RNA to a ribosome, ribosome makes the protein, protein to the either in the ER or move through the ER, Golgi is going to get it and do some modifications, and the vesicle is going to ship it. So again, I will run through all of that individually in just a second, but the picture's just blown up for you and shows you a little idea on how things are going to flow through the cell. So the ribosome. So why do we have ribosomes free, like floating in the cytoplasm, and we have some bound on the rough ER? It depends on where they're going. If they're in the cytoplasm, most of those proteins are going to stay in that cell. Okay, they're made for that cell in particular. If the ribosome is on the rough ER, then a lot of times that protein is going to be shipped. It's going to be exported out of the cell and it's going to go somewhere else. Okay. So the ER, endoplasmic reticulum, there is a rough ER and a smooth ER. I'll get to that in a minute, but in general, they process proteins. It could be glycoproteins or lipoproteins. All that means is a sugar attached to a protein or a lipid attached to a protein. Part of the ER will synthesize lipids for membranes. There are transport vesicles that come off of the ER and are used for like little vessels that move things around the cell. You can see that in the picture up top. A little bit more on the ER. So you can tell the rough from the smooth right here. You know, the rough has all the dots on it, right? And you can tell it's much flatter. The caverns in here are much flatter. And the space that is actually inside here, okay, all the space that's inside there is called the lumen. The lumen is a general term, okay? So lumen is on the rough ER, the inside tubular part down here in the smooth ER, that is also the lumen. Other organelles have lumens as well. It just kind of means the inside cavity, okay? So a little bit more on the rough ER, flattened caverns. Smooth ER are more tubular. Okay. So again, cartoon picture, microscope picture. Kind of look at both. Pause the video if you need to. Another job is to transport molecules through the cell. Kind of works like a subway. You can envision that given how all the tubes and caverns are connected. Things can go through those lumens, those tubes really well. You can move things around the cell. The rough ER is obviously involved in protein production because it has the ribosomes on it. The smooth ER is actually where the lipids are made, lipid and carbohydrate production. So if we need more phospholipids at the cellular membrane, then they're going to be built by the smooth ER and shipped out there. Next, we get to the Golgi. So the Golgi apparatus or the Golgi body, same thing. And it's right behind the ER. Okay, so down here is a transport vesicle from the ER, from the endoplasmic reticulum, and it's actually going to merge in here with the Golgi body. It's going to like ooze into it and be accepted by it and kind of merge into it, kind of like an endocytosis type thing. There are two faces to the Golgi body. You have what's called the cis face. Okay, this is the receiving side. So the cis face, this side of the Golgi, it's going to face the ER. It's going to receive the protein from the ER. Then the protein is going to work its way through these caverns, and it's going to get modified and have a lot of changes made to it. It might get a shipping label, might get moved around a little bit. Lots of things can happen within the Golgi modifications. You can add a sugar to it, you can add a lipid to it, whatever the case may be, whatever it needs. And then you have down here, you have the trans face. Okay, so the trans face, the bottom side of it down here, that is going to be for shipping. This is going to be the shipping side. Cis face, receiving side, trans face, shipping side. Okay. So a little bit more on the Golgi. Golgi has a lumen. Golgi has vesicles that bleb off of it, B-L-E-B, bleb. They just kind of like, you know, swell off and then pinch off and become their own little vesicle to move things around. You can see the proteins all in there getting modified, all the little dots. Okay, some new vesicles getting ready to pinch off. Just a micrograph of the same thing so you can see what it looks like in a microscope. Definitely a Golgi body. Definitely You can tell certain parts, feel free to label them, make sure you know how to do that. The Golgi's function is to accept molecules that the cell has made and then package them into vesicles, modify and package. You've probably heard the Golgi is like UPS. It takes in a package, modifies it, puts a shipping label on it, and gets ready to stick it on a truck to go, and the truck being the vesicle. Okay. I mentioned this earlier, you can have glycoproteins or lipoproteins, so the Golgi's putting the finishing touches on the protein, and this might be a tag for another cell to identify them. It might be a tag, so like a shipping label where it knows where to go. They can mean different things. So how this all works together. So you have, I'm going to put it all up here, and then we'll kind of, well, I'll leave it right here, and we'll kind of chat as we go. So you have the ribosome right here that's on the rough ER. It builds the protein. You can see here growing protein, which is this blue kind of tubey looking thing. So that's the protein getting built. Here's some proteins that are kind of finishing up and folding up. And this is the ER lumen, the rough ER. And then you can see this part is getting ready to bleb off and become a vesicle. And the vesicle takes these proteins and they are going to go this way to the Golgi. And notice that at the moment, the proteins inside the circles are a grayish blue. And when, I gotta get rid of my pen, when they go through here, so now they're grayish blue to start, there's a bunch of proteins. And when they move throughout the layers of the Golgi, the reason they're changing colors because it's showing being modified. And so now they're getting modified some more. And now they get into vesicles at the end, and they're gonna go wherever they need to go. So what's the fate of a vesicle? Where do they go? Well, I'm going to tell you four fates of the vesicle. Okay, so some empty their contents into the environment. You guys probably know what that's called already. Exocytosis, also known as cell throw-up, right? You're getting rid of a bunch of stuff all at one time. So yeah, cell throw-up, exocytosis. Second one, let me move my head to the other side so I'm not in the way. Second one, some vesicles are made to add new phospholipids to the cell membrane. So if you look down here at this one right here, it is adding some new lipids right in there. We still have exocytosis up there above it. Another thing that might happen is they become something, kind of like another working organelle. So they might have the proteins that are really harsh and they're used to break things down, like really strong digestive enzymes. And then they might just become another organelle called a lysosome. You probably remember the lysosome, the garbage disposal, or the wood chipper, or paper shredder, something like that. Things that are going to tear things up, right? So lysosomes are filled with very strong enzymes. And they can digest foreign invaders. They can also digest worn out organelles or possibly even the cell itself in that process called apoptosis. And then lastly, we just kind of lump everything else into something we call microbodies. There's a lot of other little things they can become. Special little vesicles that contain enzymes for specific jobs. So a couple examples are peroxisomes, which destroy toxic chemicals that build up. You might be able to actually discern what peroxisomes are going to destroy in particular. Peroxides, right? They're going to get rid of peroxides and things like that build up in the cell to help the cell stay clean and functioning. And then glyoxysomes, you might notice that gly kind of prefix, which usually indicates sugar. So glyoxysomes convert fats into sugars in plant cells. So a little thing about plants there for you. So 4 fates of the vesicle. Okay, so cell throw-up, exocytosis, replacing plasma membrane, making lysosomes, and making microbodies. Okay, so 4 fates of the vesicle there for you. And if I remember right, we are done. So I know that was a little bit longer. You are going to have 5 sets of notes for the cell unit, just to give you a heads up. So that's how that's going to go. And I will be talking with you guys soon. See ya.